The pursuit of understanding exceptional human longevity has increasingly turned toward not only those who reach 100 years or more, but also their children. Offspring of centenarians, often referred to in research as offspring of long-lived individuals, LLIs, represent a unique biological cohort. These individuals, while generally younger than their parents were at the time of study, tend to display delayed onset of age-related diseases, longer lifespans, and a suite of biological traits that suggest inherited mechanisms of healthy aging. Remarkably, many of these benefits are evident even when the offspring do not adopt particularly health-conscious lifestyles, raising compelling questions about the role of inherited biology in promoting longevity. Multiple studies across diverse populations, from New England and Japan to Italy and Israel, have documented a consistent pattern. Children of centenarians tend to exhibit significantly lower prevalence of cardiovascular disease, metabolic disorders such as type 2 diabetes, and neurodegenerative conditions like Alzheimer's disease, compared to age-matched controls. This health advantage persists across ethnic and cultural backgrounds, even when socioeconomic or environmental factors are controlled for, pointing to a strong genetic component in the inheritance of longevity-associated traits. One of the most striking biological features observed in centenarian offspring is their lower biological age relative to chronological peers. Using biomarkers such as DNA methylation clocks, researchers have shown that these individuals often have slower epigenetic aging trajectories. Their methylation patterns resemble those of younger individuals, suggesting that the biological wear and tear typically associated with aging accumulates more slowly in this population. This finding aligns with broader evidence that epigenetic stability is a key determinant of healthy aging and appears to be partially heritable. Telomere biology further reinforces this inheritance. Telomeres, repetitive DNA sequences at the ends of chromosomes, protect genomic integrity but shorten with each cell division, contributing to cellular aging. Studies have found that offspring of centenarians tend to have significantly longer telomeres than age-matched controls, and this appears to be independent of lifestyle factors such as diet, physical activity, or smoking. Moreover, telomere attrition rates may be slower in these individuals, suggesting both a higher baseline and greater long-term preservation of genomic stability. Cardiometabolic markers in these offspring also mirror patterns seen in their long-lived parents. Lower insulin levels, reduced insulin resistance, healthier lipid profiles, including high HDL and low triglycerides, and preserved vascular elasticity are common features. These metabolic signatures are closely associated with delayed aging and reduced risk of chronic disease, and many researchers now view them as core physiological correlates of inherited longevity. Importantly, these traits are often detectable decades before clinical symptoms of disease would emerge, supporting the notion that longevity is rooted in a lifelong biological advantage rather than late-life intervention alone. The immune system also appears to age more slowly in the children of LLIs. Studies have shown higher counts of naive T cells, preserved natural killer (NK) cell function, and reduced levels of systemic inflammation, particularly pro-inflammatory cytokines such as IL-6 and TNF-alpha. Since chronic low-grade inflammation, or inflammaging, is considered a major driver of age-related decline, this immunological phenotype likely contributes to the resilience observed in this group. Cognitive aging presents another area of interest. While less extensively studied than physical health, early findings suggest that offspring of centenarians may have a reduced risk of dementia and cognitive impairment, potentially due to both vascular protection and inherited neuroprotective factors, such as increased levels of brain-derived neurotrophic factor, BDNF, or enhanced neuronal plasticity. Structural brain imaging has also suggested slower rates of cortical thinning and white matter decline, though these findings remain preliminary. The heritability of these aging advantages raises important mechanistic questions. Several genetic variants implicated in human longevity, such as those in FOXO3A, APO, CETP, and IGF-1R, are found more frequently in centenarians and their offspring. These genes are involved in insulin signaling, lipid metabolism, oxidative stress resistance, and cellular maintenance. Yet, genetic inheritance alone does not tell the full story. Increasingly, researchers are exploring how epigenetic inheritance, the transmission of gene regulation states from parent to child, may shape the aging process across generations. For example, certain chromatin states or small RNA molecules may be passed through the germline, predisposing offspring to slower aging from early development onward. Additionally, maternal factors during pregnancy may play a role. There is emerging evidence that women who themselves are predisposed to exceptional longevity create more favorable in utero environments for their children, including lower exposure to inflammatory stress and more efficient placental nutrient transport. These developmental conditions can influence gene expression and organ function across the lifespan, further embedding resilience. Notably, while these biological traits are clearly inherited to some extent, they do not guarantee exceptional longevity in the next generation. Lifestyle and environment remain important modifiers. Nonetheless, the existence of this heritable advantage suggests that studies of centenarian offspring can serve as early indicators of successful aging pathways long before individuals reach old age. This insight is particularly valuable in designing midlife interventions aimed at compressing morbidity and extending health span. In summary, offspring of long-lived individuals offer a living blueprint of biological resilience. They age more slowly, harbor protective genetic and epigenetic signatures, and display delayed onset of chronic disease, even without major lifestyle changes. Their study not only deepens our understanding of the mechanisms driving longevity, but also highlights the potential for early life biomarkers to predict aging trajectories. As multiomics tools and longitudinal datasets continue to expand, centenarian offspring will remain a critical cohort for revealing how longevity is built, and, potentially, how it might be replicated in the broader population.